Okay, now we're on to the next, and this is going to be a short segment. Shouldn't take too long um, to finish up antibody-mediated immunity. And this is referred to as T-independent antibody-mediated immunity. And in this case, what's going to happen is it's a type of immunity or AMI response that doesn't require T cells, and that's why it's referred to as T-independent. Now, um, if you this response was recognized because you could see it, you could see this, you could see it in T deficient animals. It's the same type. Um, um, it's characterized by low affinity antibodies. Can be generated directly without the need for T cell activation. Most T-independent antigens have repeating subunits that are arrayed in regular geometrical shapes, and many are high molecular weight polysaccharides, um, for example, like LPS or dextran sulfate. And we'll see why. It'll become clear when we see the mechanism. Um, and then virus capsids turn out to be excellent inducers of strong T-independent AMI. Um, Particularly non-envelope capsids and viruses. And if antigen processing takes place, then isotype switching will take place, and high affinity antibodies will be induced, and that will intersect with the T response. So if we look at an antibody that expresses, you've seen this before, will express antibodies on its surface. They're bound to its surface. And you can run into antigens in which you have repeating subunits. So each one of those little red balls is basically the same antigen and determinant. Uh, for example, on a polymer, uh, they're glycopolymers that have repeating subunits, that one exactly like the other, one protein right after another. And those can engage, once those, of course, can bind to the antibody on the B cell. And what they'll do is they'll cause the aggregation of those um, antibodies on the surface. Now, most always they're IgM. And what you'll see is, you see here, is that there's a clustering. So these get clustered. Um, these get clustered. There's a clustering, so they sort of cluster together. Okay, and they form a cluster in which they interact with the antigen. And they're not going to. This response doesn't require antigen processing. What's going to happen is you're going to get proliferation of the cells, mild proliferation of the cells, and high levels of IgM secretion. So you're going to get IgM being produced. There's no memory produced. You get proliferation of the cells, but there's no memory that's produced um, with this response. So this allows you to sort of quickly get in and start making some antibody uh, to meet the challenge of an infection. And so this is another figure in which put together in which you have an antibody and it's interacting with a viral capsid, non-envelope virus, and causing a clustering of those antibodies on the surface. You'll start seeing IgM being produced. There'll be IgM that's produced right directly from the B cell. And then what can happen is, for example, if it's a virus, the virus is going to be small enough, even though it has uh, number of repeating subunits, it will be small enough to undergo internalization and antigen processing. So um, geez, what's going on here? Initially, what happens is in the sort of first step, you get this occurring in the very early recognition. So the antibody can be produced to a limited amount from the B cell, and it's not huge, but it produces a limited amount. And then as you undergo antigen processing, you'll start to get B cell activation and then IgG production. So this gives the host some ability to start making some antibody that could be pretty fairly protective to begin with. And so this would be this part would be 2 here. Okay. So if we look at how this fits together with our scheme, if you had a virus that met this credentials, or a nanoparticle that had antigen determinants every 5 to 10 nanometers, what you would see is this, of course, could undergo phagocytosis through dendritic cells and macrophages and upregulate this arm of the AMI response. But it could also interact with a naive B cell like we had before, which leads to production of IgM, and then eventually, if it can be taken up and lead to production of antibodies, IgG antibodies, by the activation mechanism that we spent quite a bit of time talking about. So that's why this part here is referred to as a T-independent AMI. Then it's coupled to, for viruses in particular, it's coupled to the T-dependent AMI, which is everything that's would be in here. Okay, so this would be TI or T independent, and T dependent would be this sort of where I've circled. Okay, so that should be relatively clear. And again, the main thing is you get initial interactions can give you antibody that's produced right off the bat. It's another reason you get IgM being produced. You can start seeing IgM produced before IgG. So just to wrap up, antibodies can be, you can remember, a take-home message: generation antibodies to an antigen depends on initial and secondary exposure to the antigen. That depends on clonal selection and somatic mutation governing the production and development of those antibodies. CD4 T cells control the B cell antibody selection and production by antigen processing and presentation that we've seen in eventual antibody. Antibodies can be used to induce antibody-based immunity to microbial pathogens, and we will see that later when we talk about vaccines. Okay, so that's it for now as a short segment. And in the next segment, we'll start talking about other elements of the immune system.